of the dawn of the oral cavity. And we all know that this dawn of the oral cavity is the tooth. And we have to care of this surrounding structures of the tooth. And this Perioshastra is the knowledge. We are gaining this Shastra. And this Shastra, the application of this Shastra is known as Perioshastra. Am I audible to all of you? Okay. So the first, this Perioshastra terminology is very good. And we have to strong this Shastra for the better application of this Shastra. So today I'm again thankful to Dr. Vivek Band for this Perioshastra. And my today's topic of my today's lecture is, good morning, Dr. KK Boss. Am I audible? And today's topic of my presentation is...
Good morning, Pavitra. Welcome to Perioshastra.
Oh, thank you, sir. Thank you. I'm just giving the terminology of Perioshastra to increase this Perioshastra. As we all know that according to Bloom's taxonomy, the application of the cognitive domain is the psychomotor skill, and this application of this Shastra is the Shastra, that is the periodontics. So the today's topic is the biofilm and periodontal microbiology. I'm going to share my screen. I think it just wait a minute. Is it visible?
Anji, Dr. Sahab.
My slides are visible?
Not on slide board. Make it slideshow, Doctor.
Okay. Now is it visible?
Anji. Bilkul.
So my topic is biofilm and periodontal microbiology. As we all know that the biological film, which is present on the tooth, a biological film, but a biological film which contains different types of biological organisms is known as biofilm. It's not changing. Yeah. Okay. Again, I think it's... my slideshow. As we all know that the periodontitis is the inflammation of the surrounding structures of the tooth, and all the periodontal diseases are the chronic multifactorial infectious diseases and affecting all the humans since the dawn of the history. I think this is moving continuously. And these periodontal diseases are multifactorial in origin. And the multifactorial means that where the multiple factors are involved in all type of periodontal diseases, and these multiple factors are microbiologic, immunologic, systemic, genetic, and behavioral. I'm not visible. Its slide is this. It's okay. Now this one is okay. So as we know that this oral cavity is a hub of all the microorganisms, as the periodontal diseases are multifactorial. And the first one is, the most important factor is the microorganisms. And the microorganisms, we can say the all types of microorganisms which are found in the universe, we can found in this the oral cavity of any human. But they live in a, close your, better don't disturb. But they live in a particular harmony. So the most important is the microorganisms. And see, this is the picture. So the first scientist I would like to mention, that is the father of microbiology, and he is also the father of microscopy. This is Antony von Leeuwenhoek. Antony Phillips von Leeuwenhoek. 340 years ago, he wrote a letter to the Royal Society. He was a Dutch, and the most important thing, he found the protozoa, different protozoas in his different rainwaters, and in the plaque of his own oral cavity. And he used the terminology of animalcules. And Antony von Leeuwenhoek was the scientist who gave the term animalcules. The second, we can say this is green, vermilion, black. He founder of modern dentistry and the father of operative dentistry. He was the scientist who gave the significance of dental plaque. The second one is W.D. John Williams. And we can the main scientist who is the John William Bill Costerton, and he coined the term that is the biofilm. A biofilm is a biological film which contains the different microorganisms. I would like to mention that Bill Costerton, he's a man of mountains. He was a microbiologist. He was a climber. He used the climbing and the rocks of Canadian rocks. And during the climbing process, he found some slippery things, that he found the bacteria is stuck in slime. This, he developed the biofilm theory, and which bacteria grows enclosed in a protective biopolymeric matrix, forming a film that is adherent to solid surfaces. And he strongly promoted the views that bacteria in biofilms differ significantly from their free-floating form. But the free-floating form is the form where they live free-floating, and when they live in a sessile or in a colony form, they differ completely. Their formation, their habitat is completely different in the biological form. And the other scientist I would like to mention, that is George Haji Shingalis. He is the current scientist who gave the theory of keystone pathogen. I would like to mention in these keystone pathogens in the later my slides. So this is the Antony von Leeuwenhoek, who gave the term is animalcules. Williams, John Leon Williams, significance of dental plaque. Black, who coined the term gelatinous microbic plaque. Weirhog, who described the importance of bacterial plaque in the etiology of periodontal disease. Losch, who's the specific theory. Sokransky, modern vision of specific plaque, and Costerton and Haji Shingalis. And the oral cavity what is the oral cavity from a microbe's perspective? What he thinks, what microbes thinks. We all know that as a microorganisms present in, they live in host by adherence to the surface. The importance of a microorganism is the adherence. We all know that fetus inside the uterus is sterile. The human fetus inside the uterus is sterile. But when it passes through the birth canal, it acquires vaginal and fecal microorganisms. And this was clearly shown by Gibson et al. And within 2 weeks, a nearly mature microbiota is established. And after weaning, when the baby is not only up to the mother's milk, when he acquires something different from the mother milks, and after the main completing of weaning, when after 2 years, the entire human microbiota is formed and comprises a very complex collection of hundreds of different types of bacteria, and Moore et al. Gave this in a general human being that 10 to the power 14 different microorganisms are present in a completely healthy human, and that is 10 times more bacteria than normal human cells. In a healthy human, there are nearly 10 to the power 13 cells, and a normal human being having 10 to the power 14 different types of microorganisms in a healthy body. But they live in a commensalism. They live in a homeostasis. And when this homeostasis changes into dysbiotic changes, then the disease occurs. In a normal human, the one thing, it's also a Moore study that bacterial population comprises 2 kilograms of the total body weight. And just imagine that the brain weight of a human being is nearly 1.4 kg, just 1.4 kg. So the colonization of oral cavity starts at the time of birth. So when birth occurs, when the baby passes through the birth canal, the colonization process occurs. And in the initial stages, facultative aerobic bacteria comes first. And from the second day, anaerobic bacteria can be detected in the infant's mouth. Now, oral cavity is basically an open growth system. Oral cavity with the oropharynx there are different types of nicks. There are different types of ecosystems are present in the oral cavity. We can classify 6 different ecosystems or 6 different nicks in the oral cavity. And these distinct ecologic determinants are present. With the first is intraoral, supragingival, hard surfaces. So maybe teeth, implants, restorations, prosthesis, subgingival regions adjacent to a hard surface, periodontal pocket, periimplantitis cases where the periimplant pocket, its crevicular fluid, the root cementum or implant surface, there's a different ecosystem. Buccal epithelium, palatal epithelium, epithelium of the floor of the mouth, dorsum of the tongue, tonsils, and saliva. These are the 6 different ecosystems are present in the oral cavity where the different types of microorganism can live in the harmony, in the normal condition, in a physiologic condition. When this harmony disturbs, the disease occurs. So the oral cavity, as we all know, that the first, the theory of infection is the adhesion, invasion, evasion. Adhesion of the microorganism to a surface is the first thing to initiate its process of infection. And we all know that they themselves host by adhering to a surface. From an ecologic point of view, the oral cavity communicates with pharynx and is the open growth system. And it maintains a dynamic equilibrium exists between the adhesion forces of the microorganisms, and a variety of removal forces are also present. We know the removal forces that are swallowing, mastication, blowing of the nose, and the tongue and the oral hygiene implements, the washout effect of the saliva, nasal and crevicular fluid, the all active motion of cilia, they all removes the bacteria. So there is a proper equilibrium between adhesion forces and the removal forces. This high turnover rate of the shedding of the epithelium act also a defense mechanism of this. But these, when the hard surfaces, they provide teeth, nails, these are the naturally occurring, different implants, or these are the naturally occurring non-shedding surfaces. And they provide the surface to adhere of the bacteria. And junctional epithelium, This is very specific type of a junctional in attachment apparatus. This junctional epithelium is very important. We all know that the ligament is a thing which adheres bone to bone, and tendon is bone to ligament. So here's the junctional epithelium. It's the only attachment process, which is the oral cavity, which is directly involved with the external environment. All different types of microorganisms have a different types of battery of virulence factors, the ability of different types of adherence activity. They act as it's a type of, we'll act as a Actinobacillus actinomycetum, comitans, Porphyromonas gingivalis. They have a different types of battery of virulence factors. They can adherence, how they invade, how they evade the host defense mechanism. These are the different types of things. We can say that teeth and nails are the only naturally occurring non-shedding surfaces. Artificial non-shedding surfaces are the dental implants, heart valves are also present. So the teeth act as also the port of entry of periodontal pathogens. In the first, when the teeth comes in the oral cavity, the first microorganism who first adhere, that is the Streptococcus mutans. And this is also a Moore study. And this Streptococcus mutans, we all know that is also known as obligate periphyte. And in the bacteria, what he thinks in the biofilm, it's not easy for a bacteria to live life solely. So they form a biological film. So life is not an easy task to anybody. We all know that we have to struggle to live. We have to struggle to survive. We have to struggle to increase our progeny. So it's a common factor to all of us, the same with the bacteria. So the first, we need the habitat. And this habitat in the oral cavity, the most important habitat is the tooth and different hard surfaces. And on hard surfaces, also on the soft surfaces. So they live in the different film. They live in a micro colonies. They need a matrix of extracellular polymeric substances that we can say this is the extracellular polymeric substance, that is EPS. This is the main matrix which provides the organized structures. Just we'll say that what is materia alba, we can remove the materia alba with a jet of water. But organized form of materia alba, we cannot remove by the jet of water. And this organization is caused by this biological film. And this biological film is just because of this extracellular polymeric matrix. And this organic constitutes of biofilm matrix is polysaccharides, proteins, glycoproteins, lipids, albumin. They can be from the host. They can be from the microorganisms also. We know that this biofilm is a dental plaque. I won't like to define this WHO definition. The plaque is a specific but highly variable structural entity resulting from this. The WHO classification, WHO definition. And Glickman is the scientist. It's a soft, amorphous, granular deposits which accumulate on surface. These are the different definitions. And we know Bowen gave a definition that dental plaque is defined clinically as a structured, resilient. Structured means that is the structural colonies are different colonies. They form a different channels in between. What they provide, the different nutrients from one to another, signaling of the different messages from one to another. This is signaling, that is all, we all know that is a quorum sensing and the coaggregation, coadhesion in the later stages. Dental plaque is general term for complex microbial community that develops on the tooth surface, embedded in a matrix of polymers of bacterial and salivary origin, that is a PD marsh from the definition of PD marsh. Dental plaques are excellent examples of biofilm communities, and they have become one of the most highly studied biofilm systems. Architecture of a dental plaque biofilm has many features in common with other biofilms. It is heterogeneous in structure. These are open fluid-filled channels running through the plaque, and different types of extracellular matrix providing a specialized environment and barrier functions. This is the picture. We can observe the different fluid channels are present. We all know that is the pellicle is the one, the important is acquired pellicle, on which pellicle there's a primary colonization occurs, then the secondary colonization occurs, then these bacterial microcolonies in the intermicrobial matrix, they are the different channels who can composed in tough extracellular matrix. They provide the different types of messaging and all the things in this pellicle. Dental plaque defined clinical structural resilience. A dental plaque is composed 1 gram of wet plaque. This is the heterogeneous mosaic model. Am I audible?
Yes, sir.
Audible?
100%. Yes, sir. You're audible, sir. No problem.
Okay. Heterogeneous mosaic model, its extreme form of water channel model it's described by Cavill. The view of such biofilm is of microcolonies forming stacks attached to the substratum at the base, but generally well separated from their neighbors. We can classify the dental biofilm or a dental plaque. It's supragingival, subgingival, coronal, marginal, tooth associated, tissue associated, and unattached. And apical parts, this is the subgingival plaque system is more dominated by spirochetes and cocci and rods. And what is the formation? See, we all know that the marginal gingivitis is the commonest. This is just because of the marginal plaque. No one of us can deprive of this. So in the oral cavity, we know that the marginal gingivitis is the commonest form of gingivitis. These are the different characteristics of supragingival, but it's gram-positive and all the things. So the process of plaque formation, we all know that the formation of pellicle on the tooth surface, first is the formation of pellicle, then the initial adhesion, attachment. These are the different host cells in dental plaques. These are the organic constituents, inorganic constituents, materials from saliva, GCF, and also from the bacteria. 80% of the plaque is water, 20% is solid. Out of this 20%, that is 70 to 80% of total solid plaque mass is the microorganisms, and rest is intercellular matrix. Other microorganisms other than bacteria are mycoplasma, yeast, protozoa, viruses. We all know that 1 cc of weighing about 1 milligram, it contains more than 10 to the power 8 bacteria. According to Linde, according to Kerenza, it's 10 to the 11. The intercellular matrix estimated to account of plaque mass, organic, saliva, GCF, and the rest. These are the inorganic contents, organic contents. Glycos in the formation of the pellicle. So the first, which is the first stage of biofilm formation, that is formation of pellicle. Pellicle, all surfaces of the oral cavity, including hard or soft tissues, they are all coated with organic material known as acquired pellicle. And we know that there are more than 180 peptides are present, including different types of glycoproteins, keratins, mucins, proline-rich proteins, stericins, extracts, etc. And this is classical studies of Hamming's study, who showed by the transmission electron microscopy that the pellicle is composed of basically of 2 layers. One is basal and the other one is then the globular layer. And this basal layer is basically very difficult to remove with any chemical. So we can say that dental enamel is permanently covered with an acquired pellicle from the moment that the teeth erupt. We can remove this upper globular layer, but we cannot remove this basal layer of the acquired pellicle. These are the substances. Albumin, probably originating from crevicular fluid, has been identified as a component of plaque matrix, etc., etc. These are the carbohydrates which are present: fructans, glucans, dextrans. The formation of pellicle on the tooth surface is that is the basal layer and the globular layer. Globular layer, we know that is nearly about 1 micrometer thickened, and this globular layer can be removed, but basal layer cannot be removed at any time. So the second phase is this. This is the clean. This picture shows that is what is the clean substratum stages in the formation of biofilm on clean, hard, and non-shedding surfaces following immersion into a fluid environment. The phase one is molecular adsorption to the biofilm formation. So first is adsorption. So this initial adhesion is the very important phase. This is the transmission: thin basal layer, thick and globular layer is 1 micrometer. And this glycoprotein is composed of more than 180 peptides. And the salivary pellicle can be detected on clean enamel surface within 1 minute after their introduction into the mouths of volunteers. This was the study of Halling et al. By 2 hours, the pellicle is essential in equilibrium between adsorption and detachment. Although further pellicle maturation can be observed for several hours. This is pellicle composed of glycoproteins, proline-rich plasma, phosphoproteins, amylase, and there is the means of different types of forces are present. In addition, there are different phases are present. Initial phase. Significance of pellicle is protective, lubrication, and it creates the nidus for bacteria, and it provides also the attachment of calculus. So this is the initial adhesion and attachment. How this on the enamel surface, this is the pellicle formation. On this pellicle, there are weak, long range van der Waals forces are present. So the first is the transport to the surface. The first stage, which is the involvement of initial transport of the bacterium to the tooth surface. And this initial transport to the surface just by, not only by random contacts, it is by random contact, it is by Brownian motion. And this Brownian motion is nearly about 40 microns per hour. This is and through sedimentation of microorganism, and on this sedimentation, they attach that with the chemotactic activity also present, and this is the formation of the primary colonizing bacteria in contact with the tooth. This is first one, we can say the transport to the surface. Then this is the adhesion, initial adhesion. Initial adhesion is the second stage in the initial adhesion. It's a reversible adhesion of the bacteria. It is reversible. And this up to reversible, it is nearly about 50 nanometers. There are long and short range. This is the Brownian motion up to 40 micron through sedimentation of microorganism, through liquid flow, through chemotactic movement. And this is the initial reversible adhesion. There is a very close reversible up to 50 nanometer. The bacterium up to 50 nanometer, it is in the reversible adhesion. But when this distance is below this is irreversible. There is strong attachment occurs. We know that is this initial adhesion. After this, the behavior of bacterial cells can be described by Durjogon, Landau, Verbe, Overbeck theory of colloidal stability. And in this theory, the total energy, which is the total Gibbs energy is the sum of the attractive forces and the repulsive forces. Then the repulsive forces and the attractive forces, the sum found this irreversible adhesion. That is irreversible binding by a bacterial cell to cell. This is 10 nanometer distance. Then 10 nanometer distance shows the irreversible binding of the bacterial cell to the pellicle. Then microbial cell surfaces are not uniformly coated with negative charges. We all know that is the negative charge which provides the positive charge, and the different van der Waals electrostatic forces occurs. Then it provides many proteins in the acquired pellicle act as receptors for the streptococci, just like streptococci, alpha amylase, acid proline rich proteins, and different stethurins, they act as the receptors for the adhesion of the bacteria. How the strong attachment occurs. Rough surface provides better protection against shear forces. So the rough surface, if the surface is rough, if the surface is rough, this attachment is more. That's why after scaling in root planing, we always tell to our student that the polishing is very, very important thing. So after root planing and scaling and root planing, the polishing is very important. Substratum surface energy provides also specific adhesion on bacterial cell. There is a complementary receptors on pellicle, alpha amylase, acid proline rich proteins, and glycoproteins. Large clumps of bacterial cells not stick well, that is removal by swallowing. So we can say when the upper layers of plaque mass increases, the shedding of the bacterial mass also occurs. This shedding of the bacterial mass occurs by removal of swallowing also. A firm anchorage is established by specific interactions. There are covalent, ionic, hydrogen bondings. Adhesions can be subdivided in two major classes. When we all know that the P. gingivitis have the major fimbriae, minor fimbriae, the fimbrial attachment. It's a type of attachments, fimbrial adhesions. This is the primary colonizers provides the new receptors for the secondary colonizers. So this primary colonizers act as the new receptors for the new bacteria. That is the co-adhesion term was given by Colin Brander. And this co-adhesion or co-aggregation formation of this mature biofilm. Co-aggregation is a direct interaction and is distinct from agglutination, which occurs when cells stick together by molecules in the solution. As we know that the initial stages of co-aggregation or co-aggregation are essentially the same as the first step in bacterial binding. So this is the, we know that the co-aggregation is it's a different types of the co-aggregation. That is the concob and bristle brush. These are the bacterium host bacterial interaction involved in adhesion. Streptococcus adhesion receptors, different types of receptors, the molecules which are present on the bacteria, and the receptors, salivary agglutinin provides a good receptor to antigen 111. Leukotoxin A provides blood group reactions. Mutants, streptococci, that is glucan binding proteins, different types of actinomyces and all the things. This is the enamel where the pellicle enamel provides the first pellicle provides the first receptors, then the primary colonizers provides the secondary receptors for the secondary colonization. And this all type of secondary colonization, we all know that the gram positive and the primary colonization, and primary colonizers gives the receptors to the, as the primary colonizers are gram positive, they use the oxygen content when they uses oxygen in their metabolism. then the redox potential reduces, then the environment becomes anaerobic. So we know that the secondary colonizers are the gram-negative and anaerobic form. This is the colonization and the classical picture of colonization and plaque maturation. We know that this is the early colonizers bind to receptors in the pellicle, then adherent cell becomes in turn the nascent surface and provides bridge for additional species. And these additional species are the secondary colonizers. This complementary sets of adhesion receptors, they provide the surfaces of the new receptors. They form the co-aggregation with the molecules in the pellicle. And these stem adhesion receptors are cellular component, that is, we know that is the heat inactivated, जो हो जाते हैं उसमें 85 degrees से 30, but the cell type exhibiting the complementary symbol receptors. So this is the classical picture which is given by Colin Brander. This is secondary colonizers. We all know this. Sokransky also gave the different types of, and this is the concave structure. Concave structure where the streptococci adhere to the filaments of different types of, when the cocci adheres to filament, it is concave. And when the filaments to filament in the test tube brush, we know. The Lisgarten and Reynolds studies, different types of well-characterized interactions include the coaggregation of Fusobacterium nucleatum with Streptococcus sanguinis, Prevotella loschi with Actinomycosis viscosus, Capnocytophaga with the viscosus, and different coaggregations between different gram-negative F nucleatum provides good towards the P. gingivalis and Treponema denticola. This is the classical study which was given by Sigmund Sokransky and Hafazi, and where subgingival plaque samples taken from the mesial aspects of different 185 adults. And each sample was individually analyzed for the presence of different subgingival species. He used the DNA checkerboard hybridization. And in this Sokransky and Hafazi study, they classically determine the 40 subgingival species in their different types of, in this study. And Sokransky, he created the different color coding systems. He used the term, we know that the primary colonizers has divided into yellow and purple, and the secondary colonizers orange, green, and red complex. And we know that the actinomyces and the different streptococci comes in the yellow and purple complex, and there we all know that the red complex, P. gingivalis, Treponema denticola, and Bacteroides forsythus, which is now known as Tannerella forsythus, is the red complex, which are the basically involved in the periodontopathogens. And these are the bleeding on probing is mainly just this because of this type of a red complex. And this color coding is just to provide the easy conceptualization of this theory. So this is the picture of this Sokransky's microbial complexes. And this picture you can see, it's a yellow, purple, the primary initial colonizers, Veillonella parvula, and we know that these are these few beneficial bacteria. There is the Streptococcus sanguinis and Veillonella parvula, who sanguis gives the hydrogen peroxide, it creates the nascent oxygen, with lethal for the other periodontopathogens, and the green, orange, and red complex. And what is this is the dynamics of the important changes in the plaque growth. What occurs during within first 24 hours, During the first 2 to 8 hours, the adhering streptococci saturate the salivary pellicle. 3 to 30% of animal surface is. These are the different stages. Rapid growth occurs after 4 to 6 hours. Josh suggested it is at as a generation time. After 1 day, the term biofilm is fully dissolved because organization takes place within it. These are the bacterial densities. And this occurs due to multiplication of already adhering microorganisms rather than by new colonizers. And thickness of a plaque increases slowly with time, increasing to 20 to 30 micrometer after 3 days. This is the first 24 hours, plaque growth is negligible. And then the ecologic shift within biofilm with transition. And we know that during night, plaque growth rate is reduced by 50%. And this is just because of the slow activity of the circadian cycle. There may be a chances of the reduction in the GCF flow. So the different studies when the plaque maturation, plaque growth rate is reduced by 50% in night, just because of the reduction in the salivary flow and the reduction in the GCF flow. These are the how it is de novo subgingival plaque formation, how the plaque formation, the time is going on. I want to describe, this is the classical study of the Lowe et al. Of 1965 on Sri Lankan tea laborers. This is the artistic impression given by Lowe in his experiment. We all know that the bacterial ecology, bacterial etiology of gingivitis using the experimental gingivitis model. In this plaque-free subjects, but they are deprived of the then mechanical plaque control. And with the time, the plaque composition changes. With the time when the plaque composition, you see this, the plaque composition changes. With the plaque composition changes during the first few days, the plaque is mainly gram-positive cocci and rods. Later on, composition is shifted towards gram-negative species, and then towards the rods and spirochetes appear more. Within a few days, a mild gingivitis ensured. This is a black line which is giving the increase. And when the 20th day, when the moment the proper plaque control is re-established, and this vertical line, the plaque composition return to the initial situation, and this is the process of gingivitis, which is the reversible condition, and the gingivitis disappears. So this is the basically given, this is the classical study, Lowe, Hilade, and it is published in 1965 Journal of Odontology. And these are the, what are the different factors influencing plaque formation? These are the different factors. There's a variation within the dentition. We know there's a more in lower jaw compared to upper jaw, more in molar regions, more in interdental areas, difficult in, this is the importance of the toothbrush. If the inflammation is more, there is a chances of more inflammation, plaque formation increases. Just because of the increase in inflammation, that means the increase in GCF. This is the increase in GCF causes the, it's shown variation with age does not affect plaque formation. But few studies showed that as the age increases, the plaque formation increases. But it's not, variation with age does not affect the plaque formation according to Linde. Spontaneous cleaning, patient eating more fibrous diet had less plaque formation. Biofilms older than 4 days do not get spontaneously cleaned. Supragingival plaque formation can also start from grooves, pericymata, and early colon. Few studies are also of the how the roughness, how the clinical illustration of this typical topography, plaque growth. Initial growth starts from along the gingival margins, then the interdental spaces, then the coronal direction, different types of fundamentally changes. See, this is the irregularities present on the tooth, crack or irregularities. Then the individualized, this is the type of an individualized plaque growth pattern. So the importance of the clean, polished surface is very, very important. Surface micro roughness, how much Curinin et al. Gave, Curinin and Lisgarten, that the surface roughness, how the more plaque growth is present on the surface, that is the threshold is 0.2 micrometers, above which bacterial adhesion is more common. This is the study by Curinin and Lisgarten, where the Teflon strip, cellulose acetate strip were used to observe the plaque maturation. How this nutrients, metabolism of plaque bacteria. Metabolism of plaque bacteria, they need nutrients from the saliva. And how this changes in the oxygen. This is the second stage. I'm forwarding this slide. The more efficient metabolism. This is the classical study of Munksgaard, and who gave that is the schematic illustration of this, the metabolic interaction among different bacterial species found in plaque and between the host and plaque bacteria. These interactions are likely to be important to the survival of the bacteria in the periodontal environment. How they defeat these competitive interactions, how they provide the different types of metabolites to the other colonizers. They provide. So this is the type of a thing of a biofilm who gives nutrients to the other colonizers, other types of microorganisms. And for now this is the thing. What is the importance of this biofilm? We all know that is the communication between this biofilm, how it occurs, the communication. And we know there's this bacterial cell do not exist in isolation. In a biofilm, capacity to communicate with each other is known as quorum sensing. And one bacteria communicate to the other bacteria is known as quorum sensing. How they use the cell phone between two bacteria, how they talk, how they show that I'm getting this more and you need this thing more. How I have all the things in my pocket, now you can get this thing from my side. This is known as sensing. And then this transfer of message, maybe transfer of gene, transfer of the things in between the different species, different cell to cell, is known as quorum sensing. And this quorum sensing is basically, we know that is this type of signaling detected dental peptide. These are two basically, either in gram positive generally peptides, they releases peptides during growth, and they provide that I have reached this condition, now you stop, I have got this thing, you can take this thing. In gram positive, it's a peptides. And we know that the second is auto-inducer. Two is this is the universal signal molecule. This is the universal signal molecule is auto-inducer 2. This is the increase the transformation frequency of biofilm. It's growing. That is the mutualistic behavior was only observed when auto-inducer 2 was present. They disrupt the gene from auto-inducer 2. They disrupt they produce that, they suggest that interbacterial communication is important for the development of dental plaque. So these interactions between dental plaque bacteria is the quorum sensing. The streptococcus, this is the same thing. The streptococcal peptides are known as competence stimulating peptides, CSP, because the major response to these is the induction of competence. The physiologic state during the cells are primed for DNA uptake and incorporation. Second quorum sensing system enclosed by the Lucknow gene, auto-inducer 2. Auto-inducer 2 is produced and detected by many different bacteria, including Fusobacterium nucleatum, P. gingivalis, Prevotella intermedia. They produce wide variety of changes in gene expressions. These are the types of auto-inducer 2. Quorum sensing is closely linked. Quorum sensing give biofilm a distinct property, which I was telling that this is the things. This what biofilm provide, what the ultimate thing which is provided by the biofilm towards the microorganism is known as antimicrobial resistance. Just imagine if a bacteria live alone, and if a bacteria live in a microbial colony in a biofilm, he is 1,000 to 1,500 times more resistant to the antimicrobial substance. So this is the characteristic of a biofilm. This provides a resistance against the antimicrobial agent. This provides nutritional status. This provides growth rate. This provides temperature tolerance. This provides pH tolerance. This provides effective concentration of antimicrobial agents outside this biofilm. This is a mechanism of resistance appears to then the bacterial species grows in the biofilm. And this resistance can cause the super resistant bacteria also. And these are the different types of things by which condition they can produce, by production of extracellular enzymes such as beta lactamase and formaldehyde lyase and formaldehyde dehydrogenase. And this provides the antibiotic resistance may be spread through a biofilm by the exchange of DNA. This DNA exchange can be just by conjugation, by the transformation, by the plasmid transformation, and one is a transposon transformation. These are the things. This is a picturization of neutralization of the antibiotics, how this beta lactamase provides and they provide resistance to the antibiotics, hydrophobic drugs like macrolides, and different types of a quorum sensing, how they can provide the channeling of that is signal compounds, that is something is coming, now you need this, something I can give to you. So this is the quorum sensing. This is the conjugation and transformation. This is a type of a conjugation where this is the super resistant bacteria can form. See, this is the class. This is a picture which it's a sex pili. It's a conjugation. It's a type of it's a sex pili, and the production of a new variant. And just we are facing a type of a thing which we faced in the last year. There's a pandemic, and I quote the terminology. It's an endemic, which has the passport is known as pandemic. So this is just because of a new variants are coming of this COVID-19 coming nowadays, such type of things. So just like this is the conjugation, transformation, plasmid transfer, transposon transfer, and all the things are different things. And one more important thing is bacterial transmission. See, this is a bacterial transmission is from pathogen from one locus to another is an important aspect of infectious disease. From one arch to another, from one site to another site. This is one is an intraoral translocation, and this is a horizontal, the passage of infections between individuals. But vertical is the transmission is known as the from parent to the progeny. That means that is type of aggressive periodontitis coming in the second generation. Or the other thing, the other things that is known as vertical means from parent to offspring. So parent to progeny is known as vertical transmission. That is the most, I can say, this is the very not so, but very detrimental thing. This is the vertical, and we have there is two studies here I would like to mention. One is curinin and one is the second group is leuven. And this is the very common in our daily routine also. Curinin use the treatment strategy of one stage full mouth disinfection. That means if a non-surgical periodontal therapy, full mouth disinfection in one sitting. That is all scaling, root planing done in single sitting. That is the curinin. And leuven group is the classical group when approach of a standard therapy, root planing is scaling in one quadrant, and then they allows. But this curinin study showed that if you are doing all removal of etiological factor, then etiotrophic phase, phase 1 therapy, if you're performing all the removal of all the etiological agents in a single sitting, there is a chances of the horizontal transmission or the translocation from one locus to another locus is reduced. So this is the translocation can be saliva act as a medium. So the full mouth disinfection has come into picture. Full mouth scaling and root planing within 24 hours to reduce the number of subgingival pathogenic organisms. Subgingival irrigation of all pockets with 1% chlorhexidine. Tongue brushing with an antiseptic. Mouth rinsing with an antiseptic. These are the changing views of plaque hypothesis. These are the different, we know the different hypotheses of the plaque hypothesis. We know the disease, earlier it was we know the non-specific plaque hypothesis where it was in the mid 1900. This is a golden age of microbiology. The periodontal diseases were believed to result from an accumulation of plaque over time. That means the more plaque, the chances of more disease. This is the non-specific plaque hypothesis. The theory, it's a that is known as the amount of plaque depends on the condition of inflammation or the disease. That's the evidence of periodontitis. But as the time changes, it comes towards a specific plaque hypothesis. That means the specific plaque is detrimental. Specific plaque is detrimental. Not all plaque are detrimental. Then comes to the ecologic plaque hypothesis. It's also given a Marsh. An ecologic plaque hypothesis and ecologic plaque it's a Marsh and co-workers showed that this is unify the existing theories that the attempts the ecologic plaque both amount and the specific microbial composition of plaque may contribute to the transition from health to disease. We know that disease is a game of a cytokine, and disease is a game of a cytokine. We know that dis of the ease is disease, that the deviation from the comfort is the disease, and how the disease occurs. When these microorganisms live in harmony, they live in the symbiotic region. They provide health. When this symbiosis changes into dysbiosis, the disease comes. So the dysbiosis theory is the latest theory of polymicrobial synergy and dysbiosis. So the importance is the dysbiosis. So we all should think about how we can live in symbiotic phenomena. And this is a very natural phenomenon, and it happens to all of us also. So if we live in harmony, if we want to live in healthy condition, we have to live in harmony. And it is a common in all the scenarios. In a public also, if we want to live in harmony, we live in the symbiotic, within the symbiosis. And when the symbiosis changes into dysbiosis, the disease occurs. So this is the picture of reduced plaque, how the health changes into disease. Stress, environmental changes, ecologic shift. Stress, that is the thing. That is, the disease is a game of a cytokine. How they reduced inflammation. Today's article in the newspaper of Times got it's at how much importance of a sleep, 7 hour sleep, the reduction of stress. It's an important point. Reduction of stress means the reduction of level of inflammatory cytokines. So the level of inflammatory cytokines is reduced. Anti-inflammatory cytokines increased. So if you want to reduce the inflammation, reduce the stress. The same thing in the oral cavity, same thing in your human body, same thing in all the microbiota, same thing with the environmental changes. You have to, your environmental factors should be very cooperative to all the things. Just same in the human body, just same to yourself, just same to the oral cavity. The same thing is the ecological shift of the, same thing in the ecological shift of the bacteria. If we are changing the stress and environmental change, the ecologic shift changes. When the ecologic shift changes towards positive to negative, just see the term of negativity. When the negativity increases, positivity reduces. And when the positivity reduces, the same thing, positive bacteria reduces, the negative anaerobes increases, the chances of the inflammation increases, the chances of the disease increases. Those are the very common things. So this gradual shift, am I audible to all of you, please?
Anji, Dr. Saab, you are audible.
Okay, then thank you. कुछ समझ नहीं आता कि क्या बोल रहे हैं, किसके सामने बोल रहे हैं.
जी.
So this is a very important.
हाँ जी.
So this is gradual shift of dysbiosis may result in chronic. So this is the gradual shift in microbial things. So the latest Haji Shingales is the dysbiosis thing. Dr. Ban, I am visible to you?
हाँ जी, Dr. Saab, बिल्कुल.
Okay. So हाँ, मैं तो अब देख रहा हूँ शायद. So this is the main thing is the importance of the ecology, stress, and the symbiotic relation. So symbiosis is very important, as I'm telling you in my earlier slide. So reduce the stress, reduce the inflammation, reduce the cytokine level, reduce the unpleasant environmental factors. As we know that the environmental factors, that is we know that smoking is detrimental to the different periodontal diseases. So the reduction of environmental factors is important. It's not changing, keystone pathogen. So this is the beyond the red complex. This is the important article by Haji Shingales, beyond the red complex and into more complexity, the polymicrobial synergy and dysbiosis. So in my personal view, this dysbiosis is the main etiology of all the diseases, not only periodontal diseases, just like all the chronic diseases are multifactorial in region. And we know the multifactorial means the microbiologic, immunologic, systemic, genetic, and behavioral. So we all should change our behavior. We all should reduce our stress to increase the immunity. That means the immunologic factor should be more important. Reduction in microbiology, reduction in microbiologic factors. So the importance is these 5 things: microbiology, immunology, systemic, genetic, and behavior. We cannot change our genetic thing, but we can change our environmental factors, our stress factor, are microbiologic factors. We can reduce, we can change. So as a periodontist, we can prevent the different types of periodontal diseases to develop towards we can diminish the severity of these type of periodontal diseases by such type of factors. Thank you.
Thank you so much, sir. Am I audible, sir?
Yeah, sure.
Thank you so much, sir, for such a detailed, comprehensive, and power-packed lecture. We all understand, sir, how challenging it is to cover the entire periodontal microbiology in a 1-hour schedule. But it is you, sir, who always makes impossible things possible. And sir, we would like to inform all the participants that this lecture will be on YouTube also. So this is such a wonderful presentation. You can take notes of it out of it from YouTube, and there are so many keywords which you can Google on and increase your knowledge. Now I would like to invite Professor Dr. Vivek Benz, sir, the head of the Department of Odontology, Saraswati Dental College, Lucknow, for his words of appreciation. Over to you, Dr. Vivek, sir.
Thank you, Dr. Pavit, for the wonderful lecture and the way you actually, you have actually carved out time from your busy schedule, and in 1 hour you complete the entire microbiology, which is the students can actually make notes.
It's not possible to complete its periodontal microbiology in
that topic. Definitely.
It's a huge topic.
But you have given the salient feature of this topic, Doctor.
Conclusion, just only, absolutely.
And that helps the student in understanding and writing the answer also, and understanding the concept. So thank you very much. On behalf of Saraswati Dental College and on behalf of Department of Odontology, I am hereby sharing the certificate of appreciation with you, Doctor. We will send this certificate via email. And thanks a lot. Thanks all the participants, all the audience. Dr. Manish Khatri was there, Dr. K.K. Gupta was there, and other eminent faculty members were also there besides postgraduate students. So thank you, Doctor. Thank you very much. Thank you.
Thank you. Thank you, Dr. Benz. Thank you.
Thank you, Doctor. Thank you, Jay.